Hello, everyone, and good morning, or, well, regardless, good afternoon, depending on where you are in the world. Just quickly want to do a sound check. Can everybody hear me? Looks like the mic is working. Excellent. There we go. Okay, guys, today will be the first formal day of Module 2 on biology. Now, the emphasis for Module 2, the emphasis for the biology segment, will be not just on the fundamentals, the action of cells and cell membranes and systems, etc., but also trying to highlight a lot of places within the body and especially within sports science, nutrition science, and elsewhere, where you see a lot of these basic principles in play. Today we will be discussing the cell membrane, particularly talking about channels, transporters, and really seeing how a lot of these kind of interplay within the body for some processes that we actually, most of us in the field, speak about on a daily basis. So without further ado, now, going over the basics of the cell membrane, most of you probably remember from high school biology, and we'll see an image on the next page. The cell membrane is what's known as a phospholipid bilayer, and we'll be looking at the structure in just a second. It is important to remember it is permeable to certain lipids, small lipids, gas and alcohol, as well as water. It is impermeable without assistance to pretty much everything else. And of course, we'll see why that's critical soon. Now, we're going to talk about a couple different concepts, a couple different ways in which transport across the membrane takes place. But first, again, let's look at the cell membrane itself. Now, what is important to note about the cell membrane, as most folks know, or again, will vaguely recall, is that each side of the membrane is a phospholipid, which has a hydrophilic head, which is polar, which is, of course, attracted to water, and a hydrophobic tail, which pushes away from water. Now, what that means is that the double bilayer is, of course, able to be soluble in water on the outside, but it maintains integrity in any kind of fluid environment. So again, just have another look at that. Now, important, when we talk about the cell membrane itself, it is not completely waterproof per se. Water can diffuse across it, again, alcohol and especially gases. Now, the concept of gas diffusion is extraordinarily important, of course, because it's important to note, especially things like contracting muscle cells, etc. Gases and every other substance loves to follow its concentration gradient across a semi-permeable membrane. As a cell begins to contract and utilizes oxygen and builds up carbon dioxide, the cell attempts to get rid of carbon dioxide. It diffuses outside of the bloodstream, and oxygen diffuses from the bloodstream into the cell. This is a passive process. It takes no energy. It takes no active transportation. Same thing with water. Now, water diffuses back and forth across the membrane. The concentration of solutes in the solution or substance which the cell is in makes a major difference. Now, if a cell can take in unlimited amounts of water, ostensibly, this is why maintaining a proper osmolarity, proper sodium levels, etc., in the bloodstream makes such a difference. One example I want to go into here. Now, there is the notion that drinking salt water will kill you. And of course, you say, well, why is that? Of course, taking in too much salt, obviously sounds like a bad idea. When you drink salt water, if you are starving on a desert island or in a boat in the middle of the ocean, and you start to drink salt water, the salt water goes into your gut. And now let's think about, first of all, what will happen across your intestines if your gut is filled with very, very concentrated salty fluid. Water that is so high in sodium, so much higher in sodium and salt and other particulate matter than your bloodstream. Well, if your cells, especially the ones lining the gut, let's just take this as an example, want to maintain equilibrium, what's going to happen is that all the water from your bloodstream is going to want to essentially equalize, or there's not going to be any effort to bring water from an area of higher salt concentration to an area of lower salt concentration, i.e. your bloodstream. Water is going to want to go the opposite way. It's going to want to go from your body into your gut. basically to help balance out all the huge amounts of sodium in there. What that's going to mean is that salt water is actually dehydrating to your body. Excessively salty water will actually sit there in your gut and pull water out of your bloodstream. Now, there are a couple different mechanisms by which your body can actually forcibly absorb that sodium, which we're going to talk about in a moment. It usually requires the presence of A, more water, and B, glucose, etc. Assuming you even could, and assuming that your bloodstream suddenly became very, very hypertonic and has a lot of salt in it, suddenly you're going to see the same action between your cells and the blood itself. Water is going to passively diffuse out of your muscle cells, etc., into your bloodstream to buffer the large amounts of sodium. What's going to happen in that case is that everything from muscle cell action to brain cells to organs, etc., are going to slowly fail as all the water flows out of the cytoplasm into the bloodstream. So that is why salt water kills you. Simple properties of diffusion, osmosis, and your body essentially needing plenty of other substrates to excrete all that sodium. There we go. Now, here is the cell membrane again with an example of two different proteins in it. Now, you can see on the right, the first protein up there on the picture on the right, which comes from the OpenStax textbook, is what's called a channel protein. Notice how it appears to be just an open tube, essentially, for compounds, for little particles to go through. Now, unlike passive diffusion, which is through the membrane itself, this is, well, sorry, unlike, essentially these channel proteins will allow any compound of the right size, which is typically small molecules, to actually go through them. Now, the body can control these. Channel proteins can be temporarily closed by certain action of certain activities, action of certain enzymes, certain ligands, etc. The bottom picture is of a carrier protein. Notice that the carrier protein actually brings a compound into it, it brings a molecule into it, closes up, and transports it through to the other side. Now, what we are talking about here, these are passive mechanisms that we're discussing at the moment. Now, when we say passive versus active, we basically mean any passive system does not require energy. If you look at those channel proteins, they're basically little tunnels that small molecules can go through. Carrier proteins, they don't require energy to activate. You would think they might, but what typically happens with a carrier protein is that they are essentially compound specific. A certain molecule comes towards the carrier protein, is taken in, and the carrier protein, certain sites on the carrier protein recognize the molecule, and they undergo a conformational change. Oh, looks like we have a question. Oh, this is interesting. You guys don't appear to be able to see the slides. There we go. Can you see them now? Okay, there we go. Yeah. Well, that was weird. I just simply shared it again. All right. Well, it's okay. You didn't miss anything too pretty in the last one. Let me back up for a second. Here is the phospholipid bilayer, which I think most of you folks have seen. Can you see it now? Please tell me yes. Okay. I tried to get the little dashboard out of the way, and I think when I did that, I hit it. Okay. Here is the cytoplasm. Here is the passive diffusion. And here we go. Here back to carrier proteins. Okay, so again, looking on the right, here are the channel proteins that I spoke about that you could see look like tubes, and down at the bottom, the carrier proteins that actually pull things across. I'll give you a second to look at that. Here's the phospholipid bilayer. I think most of you can see my little pointer here, and you can see the dual design. Now, okay. Channels, now, again, one thing about channel proteins especially is they distinguish the molecules that can go through them basically based on size. These are channel proteins. Now, I said carrier proteins operate by recognizing a certain molecule and actually changing conformation and basically pushing things through or allowing things through. Channel proteins have no such similar action. Channel proteins are basically just open, and if something is the right size, it can go on through. Now, important to note also that If the cell, for example, wants to change the gradient of something, remember everything goes through the processes of kind of, going across the concentration gradient and maintaining a kind of an isotonic environment. If a cell wants to create a voltage or an action potential, in other words, if a cell wants to increase the quantity of certain, like say, positively charged ions inside the cell, it would have to utilize gated channels to effectively build up these ions within the cell so it can then release them to initiate an action potential. I'm all paranoid about my screen not showing here. Okay. Now, I said channel proteins, the open channels, really only distinguish based on size. Carrier proteins, as mentioned, are what's called stereospecific. In other words, they look for a given configuration or portion of a given molecule to recognize any sort of substrate coming through. Now, carrier proteins, channel proteins are always passive. Again, just think of them as a small tunnel. If you're small enough, you can fit through anytime you want. The cell can eventually close it off if it decides to, but for the most part, things just flow in and out. A carrier protein can be passive or active. Now again, passive. Say a molecule comes across, binds to the outside. When it binds to the outside, basically just think of the shape of the molecule itself touching certain parts of this protein and causing the protein to warp ever so slightly and basically switch around and squeeze the molecule out the other side, whether it's inside or outside the cell. Now, passive examples of passive carrier proteins, as you can see, glucose uniporters on the left. And we'll get a couple of examples of each one. Now, what is important to note with this, a uniporter is a carrier protein where it brings a single molecule through in one direction. A synporter brings two molecules in at the same time, and an antiporter basically runs it as an exchange. Now, we'll see examples of all of these, and actually the rest of the segment is going to be discussing a couple of cases, systems, mechanisms you're very familiar with, where all of these exist. Now, active carrier proteins, which we'll also get into a moment, require ATP. Now, you can see an active protein would be, say, the proton pump in the GI tract. Now, why would that be active if you think about it? Well, a lot of carrier proteins can actually move ions back and forth, and move molecules back and forth. The proton pump, for example, say you want to really increase the acidity or really drastically drop the pH of a certain internal organ, like your stomach, for example. Maintaining that extreme concentration of hydrogen ions, bringing the pH that low in your stomach is energetically costly. And consistently pumping those protons out into the GI, into the stomach, is extremely energy intensive. So that is an active versus passive. Okay, an example uniporter, GLUT1. Now, that is one of the glucose transporter, here, let me take a look at A, is one of the glucose proteins that is located on the surface of cells. Now, it's located on the surface of beta cells, muscle cells, et cetera, et cetera. These are the carrier proteins that bring glucose into the cell. Simple as that. Example symporter, back here, the red one. SGLT1 in the gut. This is a very, very crucial symporter that I've discussed quite a bit. And if you look in the gut, here, see if I can pull up a picture for you here. By the way, this diagram is wrong because it's a symporter, S-Y-M. So that's from your book, don't blame me. Now, everyone familiar with hydration drinks? Some of you folks have seen, well, let's really talk about the history of hydration drinks just for a moment. Now, the concept of a hydration drink, a lot of it was originated when we talk about oral rehydration therapy. And some folks who are familiar with certain products that CHP has played a role in knows how often the ORT, or oral rehydration therapy guidelines, are utilized by us in recommending different types of hydration beverages. Now, oral rehydration therapy was originally developed as a cheap and effective way to rehydrate mostly children, looking at diseases like cholera, etc., which in a lot of the developing countries kill via dehydration. Basically, what happens is they just cause uncontrollable diarrhea, just way too much water in the GI tract, and cause just dumping, and eventually children especially will simply die of dehydration. Oral rehydration therapy were packets of salts and small amounts of other compounds that were designed to help the body forcibly absorb more fluid. Oral rehydration therapy in clinics can often, assuming a fully functioning GI tract, can often be as effective as intravenous rehydration. Now, intravenous rehydration, of course, you can do boluses, you can do huge amounts of, you can do huge flow rates in the case of massive trauma. But if you're talking about effectively an otherwise healthy individual who is not experiencing major trauma, in other words, you haven't just lost a limb or something, properly balanced oral rehydration therapy in the absence of vomiting will rehydrate you as quickly as an IV. So, for example, if an individual is running a race, or you may see this at the end of something like Ironman triathlons, in the medical tent, they often have IV therapy available if an individual is suffering from potential hyponatremia or any sort of dehydration. The one thing they stipulate typically is that you may only receive an IV if you are actively vomiting. The reason for that is in the absence of vomiting, oral rehydration therapy is just as effective. Now, why is oral rehydration therapy, or those particular compounds, why is it so crucial that they contain not only electrolytes but glucose as well? As an anecdote, the first marathon I ran had a lovely new sponsor, which I think lasted for 2 years before the company folded. That was a low-calorie hydration drink. It was a sugar-free hydration drink, which was an absolutely absurd concept because sugar itself is critical for helping the uptake of sodium. Because remember, hydration isn't just getting water in the system, it's also balancing out and maintaining isotonic blood concentrations of these solutes, because otherwise the cells simply can't absorb. And absence of electrolytes, of course, loss of sodium causes loss of muscle contraction, etc. So hydration is more than just water, as we know. But why is glucose important? Well, one of the symporters inside the gut, SGLT1, is a sodium and glucose symporter. In other words, it operates and it takes in sodium in the presence of glucose. By leveraging that, by including glucose in with oral rehydration therapy, or by including small amounts of glucose in what you are drinking, it actually activates these SGLT1 symporters and facilitates the uptake of sodium. This is absolutely critical, because the presence of glucose can help a stomach, or can help your small intestine take in significantly more sodium and significantly more water because of that than solutions that do not have glucose by the activation of these symporters. There are other symporters as well. There are actually symporters, sodium symporters, that actually also bind with amino acids. Certain amino acid symporters, if you want to look them up independently, also operate under similar concepts. That is why I believe it's the addition of alanine to certain oral rehydration therapy further aids in sodium and fluid uptake, because alanine is also it's an alanine sodium symporter in the gut. So same sort of thing there. Very critical concept to understand. A symporter does not activate without the presence of both substrates. Okay, and the last one would be an antiporter. This A and B. One thing goes in, one thing goes out. Now, an antiporter would be something like the calcium pump in cardiac muscle, where essentially cardiac muscle, anytime the muscle activates, the muscle contracts, it is constantly pumping one ion in and one ion out. And it's just an even exchange. And basically, this channel continuously pumps these things against their concentration gradients. Basically, you maintain even action potential, you maintain even polarization. And then when the muscle contracts, you depolarize. This is something we'll go into a lot more later when we discuss skeletal muscle. But typically, those are antiporters. So they only activate, again, if each compound is present on the other side. They will not pump one way. Okay, here's an example again of the sodium-potassium pump. Now this is an active transport. And you can see that the way these active transporters actually work, they actually utilize the ATP molecule itself, the phosphate on the ATP molecule, to actually change the configuration and cause that modification, cause that, I don't even know how you want to describe the concept. Things go in one side, the ATP shuts down the other side, and essentially that's where the conformational change comes from. So this is energy intensive transport, which again, we'll talk about a couple different examples of this. But basically one of the ways to think about this is anything that uses any kind of action potential or anything that is pumping ions into a gradient where, again, the objective is to create a very high concentration on one side or the other is probably active transport. Okay, now here's an example of something that is discussed quite a bit and something which a lot of us talk about that is almost based purely on the understanding of various channel proteins, various carrier proteins. Let's talk about insulin, how insulin works. Going from start to finish, everybody knows when you eat any amount of carbohydrates, or really honestly any energy substrate, your body first releases a small amount of insulin simply by the presence of the smell of good food, or the anticipation of food. Now, this is something that is discussed quite a bit, and some of you folks may be familiar with something called the cephalic insulin response. This is the notion when people talk about, oh, well, just taking in any kind of sugary drink, just taking in Diet Coke raise your insulin levels. If anybody tells you that Diet Coke does not cause an insulin release, they are lying to you. Everything with any amount of flavor or even a good smell, an appetizing smell, can cause a small spike in insulin. That is basically a fact, but that is not the mechanism we're discussing here. Once glucose actually makes it into the bloodstream, or again, any energy substrate makes it into the bloodstream, this causes insulin to be released from the beta cells in the pancreas. Now, one of the most interesting things in looking up this mechanism is you'll see a lot of question marks even in the most recent scientific literature, which goes to show that even the fundamentals are still very much kind of open for debate. Science is still very much the most the most sincere, the most well-founded concepts in science are still called theories. So glucose in the blood binds to the GLUT1/2 receptors in beta cells and enters cells. In some places you'll read it's the GLUT2 receptors. In other places you'll read that GLUT2 receptors are mostly found in rodents and it's GLUT1 in humans. Again, you'd think we almost don't really know which is which or which one is primarily active. Either way, so glucose in the blood binds to these receptors and enters the cell. Now, if you'll remember, GLUT1 is a uniporter, and here we are. Yeah. It is a passive uniporter. Okay, so no energy required to get those glucose molecules into the beta cell. Okay. So here's your beta cell in the pancreas, and glucose floats by in the blood and gets pulled in by these specific receptors. Okay, cool. Glucose metabolism generates ATP. All right, so what matters here is the ATP, adenosine triphosphate to adenosine diphosphate ratio rises. At this point, a couple things to note. First of all, the objective here, or the result, the signaling here is ATP. Has nothing to do with glucose anymore. This is why certain foods that can be processed for energy, for ATP, also cause a small insulin release. Because at this point, we're no longer talking about glucose, we're simply talking about ATP, right? So beta cells don't really care so much that it's glucose, they just care that suddenly the ATP concentrations inside the cell start to rise. Now, that ATP ratio, as it rises, it closes a potassium channel. Now, remember how a lot of channels just are, like I said, they're basically open most of the time and just allow small molecules like potassium to flow in and out? They can also close. ATP actually binds to the inside of this channel and shuts it down. It snaps it shut. what starts to happen is potassium builds up inside the cell. Now, again, so far, now this doesn't actually utilize the ATP, by the way, not yet. Now, as the cell starts to become less negative, or less negative is another way of saying positive, remember potassium ions are indicated as K+. These are positive ions. So the inside of the cell, which is normally slightly negatively charged, suddenly becomes less negative. Depolarization is the next step, which is basically where suddenly channels open and ions flow back and forth very quickly to restore your basic base charge. As it depolarizes, a voltage-gated calcium channel opens. Now again, this is another channel that just allows small calcium, Ca2+, ions to flow back and forth. Normally these are closed until depolarization happens. Suddenly these open up, calcium flows into the cell. Calcium itself, external calcium, is a signaling compound which causes little vesicles inside the cell, basically little transporters, little cells within a cell. And again, we can discuss these when we discuss organelles, to actually head to the surface, to basically break off where they are on the inside of the cell, head to the surface and release insulin into the bloodstream. So this is all a relatively, and again, this is still theoretical. There's still quite a few things we don't know, but this is all insulin. The entire process of insulin release has to do with the operation of these channels. So again, if you want to look it up as well, it's very interesting still to see how much of this is considered theoretical, but it's also very interesting to see that this is something as simple as it's a demonstration of uniporters, it's a demonstration of channels, it's a demonstration of how channels can be shut, and how this ionic switching gradient is fundamental to something that we all talk about, insulin release. And what we'll talk about in one of the papers is how various conditions can actually alter this simple process. But again, a few critical takeaways. Remember, it's the increase in ATP that actually matters when it comes to the insulin release. And this is, again, almost a purely ionically driven process. Okay, now here's another picture of the cell membrane. They're your channel proteins. They're no carrier proteins picture, but we can just picture those as channel proteins that operate one way or another. Now, you can see the little cholesterol molecules inside the cell. They are a critical part of the cell surface. Now, any cell itself, this is why cholesterol is one of the places cholesterol is contained. It exists in every single cell. You almost can't get away from it. Certainly not while you're eating any sort of animal protein, because it's an integral part of the inside of any cell, of the cell membrane, etc. Cholesterol plays several functions. It helps maintain membrane fluidity. Now, the phospholipid bilayer, of course, there are all these other proteins in there that all have various properties and change how the cell membrane itself operates and moves and everything else. But cholesterol has an interesting property where the temperature, the outside temperature, actually affects how cholesterol effectively binds and controls the fluidity of the membrane. There are certain types of cells where they will actually get somewhat viscous under high temperatures. Now, none of this is really of much note in humans, but there are a number of prokaryotes, some of the cells that live around, some of the bacteria and other, archaea that live around, like say, the superheated underwater vents, where it is the various presence of compounds like cholesterol in the membrane that allow for the cells to maintain integrity in those high temperatures. Interesting to note is that it is not cholesterol in certain other species. For example, fungi. Some of you may recognize ergosterol as a drug target for antifungals. This is, if you know vitamin D, vitamin D utilizes this as a precursor, well, vitamin D2, which is why, now, if you look at it, it says also this is not found in humans. This compound itself is what fungi use in place of cholesterol. If there are compounds like certain antifungals, clotripizole, which some folks may recognize as being like, say, an active part of antifungals like anything there for athlete's foot or anything else. What that compound does is it blocks the synthesis of this compound. Now, by blocking the synthesis of it, that doesn't hurt humans. It also doesn't hurt most bacteria. It only hurts organisms that utilize. Sorry, I lost my volume there for a second. What was I saying? Oh, yes, it only hurts organisms that utilize compounds other than cholesterol in their cell membranes. So, important to recognize here, and I think the takeaway from this section that's important, is a lot of compounds in cell membranes may very well be cell type specific. In other words, there are numerous types of carrier proteins. Let's go back here for a second. Yeah. There are numerous types of carrier proteins. There are numerous types of glycoproteins, glycolipids, etc., that are very, very specific to different types of organisms, to fungi, to prokaryotes, to bacteria. There are certain things that are inherent in cell walls, which we're not really going to get into too much because it's not really relevant. But a lot of drugs, a lot of compounds preferentially target these. So this species-specific and, in fact, entire class-specific compounds is one of the ways that a lot of antibiotics, antifungals, etc., actually function. For example, if there are certain carrier proteins and channel proteins, or again, compounds, that are only found in one particular type of organism, any sort of drug that blocks the expression of these things or the generation of these things is going to effectively remove it from the cell membrane of these bacteria. So we may actually do a paper on antibiotics and discuss a lot of these kind of organism-specific proteins and organism-specific subcompounds. Okay, any questions on any of that so far at all? Just want to give a moment. I realize I've been blabbering fairly quickly. Okay. Oh, wait, one question. Oh, yes, here we go. Somebody wanted to take a look at the first slide again. Yes, this entire presentation will be up, and all of this should be in the segment of your book that we had the recommended reading for this time around. Okay. Yes. Okay, yeah. The other question now, for hydration, the proper ratio, now, yes, it very much depends on the presence of other molecules in the gut. Everything you eat, everything that is in the intestine will, of course, contribute to this, to this kind of concentration gradient, presence of solutes in the GI tract. This is typically why oral rehydration therapy, and this is actually a very good question. This is why oral rehydration therapy is, or let's put it this way. A recommendation for athletes will often be to drink diluted Pedialyte and the like. And the reason for that is the oral rehydration therapy is designed to be as close to isotonic as possible. The idea is to minimize the need for additional water to basically facilitate the uptake of electrolytes and basically maintain an even concentration gradient as possible between what is in the gut and what is in the cells in the brush border lining of the intestine and what is in the bloodstream. If what is in the gut is hypertonic, and this is actually something that greatly impacts speed of digestion as well, by the way. But if what is in the gut is very, very salty, this will actually tremendously slow down the rate of digestion for a number of reasons. Again, if you think about it, if like mentioned with seawater, if you have a lot of salt in the gut, what's going to happen is that basically the body is going to not, or is going to slow absorption of a lot of these molecules. Like, for example, we were talking about sodium transporters, symporters, etc. A hypertonic solution, it is very rare for solutes to flow. They don't easily flow from a hypertonic solution like what is going to be in the stomach or what's going to be in the intestine to the bloodstream. A lot of the food in your GI tract needs to basically be broken down and diluted, especially by water, before it can be easily transported into the bloodstream. Now, if you are an athlete, or if you are working with an athlete, and like, let's take a practical example here. Let's just say they did a weigh-in. And what is absolutely, absolutely critical is maximizing the rate of hydration. For a weigh-in, they are typically going to be in a fasted situation. Now, what would be important for them is if they drink something that is purely isotonic, like a pure Pedialyte, that is the best single choice for them until the bloodstream actually becomes, or basically until they start to rehydrate. That is critical step number one. Now, if you mess that up either way, if that solution that's in the gut is either hypertonic, in other words, too much salt, too much food, like what typically happens after an IHOP binge, or if it's too hypotonic, in other words, if they're just drinking plain water, their rate of absorption of water and electrolytes is going to be significantly slower. So that is why the recommendation for a fasted athlete is always pure Pedialyte or pure oral rehydration therapy / isotonic solution. Now, if you have an athlete who is not fasted, chances are the solution that is in their gut is slightly hypertonic. In other words, there's already going to be the presence of glucose, presence of salt, presence of other molecules, other ions in their GI tract. If that is the case, drinking something that is isotonic is actually just going to add more solute. What you want to do is dilute that slightly. So if you have an athlete who's running a race or anything else, and let's just say you have an individual you are working with who is currently dehydrated but has food in their stomach, you don't want to give them pure Pedialyte because, again, it's already hypertonic in their GI tract. You want to give them slightly diluted Pedialyte or something along those lines. And that's going to help balance out that osmolarity. You don't want to give straight water, because chances are they may not have enough sodium, they may not have enough potassium, they may be lacking some fundamental electrolyte, but you will definitely want to dilute that somewhat. In fact, you dilute it more and more as they have more and more food or anything else in their GI tract. So this is something where, kind of maintaining that always has to be calibrated based on existing molecules in the GI tract. And it's, of course, worth looking at what they've had and saying, okay, is there, looking up those different kinds of symporters and realizing that different molecules, different macronutrients can actually facilitate the uptake of sodium. So hopefully that clarifies that a little bit. Okay. Any other questions on any of this so far? Okay, yes, insulin. Oh, God. Well, that was actually a question that just came in on Facebook. I'm so glad I'm looking at that. Okay, yes, the insulin system, insulin release. Yes, technically, large amounts of fat could technically cause insulin release as well. Because remember, it's the simple presence of ATP. When you look at this entire process, again, ATP is kind of the real trigger for a lot of this. Glucose is simply the fastest available energy substrate to kind of kickstart this process. Now this is all still theoretical. It's theorized that remember, it's the ATP to ADP ratio. So if you're, a cell that's simply regenerating ATP from like creatine phosphate, for example, creatine doesn't affect insulin release. And the reason why I've actually had that question way in the past is the question was, if creatine phosphate or a similar compound that quickly replenishes ATP, if that causes levels of ATP to rise, would that cause more insulin release? And this was actually a question that was posed ages ago. I forget the exact context, was the question that creatine would cause insulin resistance via this mechanism. But that likely does not take place because ATP resynthesis from creatine phosphate is typically still below the kind of equilibrium level needed for this whole insulin pathway here. Does that make sense? Okay, so the segment we're going to go into next, and I hope you're able to keep up with the reading on this, is going to be all of the cell organelles. Discussing a little bit, mitochondria is going to be the main one, discussing how they actually function, and really discussing a lot of topics around that. What does mitochondrial profiles mean? What does it take for a mitochondria to proliferate? And a lot of those topics that will come in very useful when discussing things on aerobic metabolism. So, by the way, most of my presentations are going to be between 40 and 45 minutes. There may be a few that go on a little longer, but again, remember, if you have any questions after you look at the reading, please feel free to bounce those over ahead of time as you're going through it, and you have especially theoretical questions. In other words, are there any concrete examples or mechanisms you can think of where this happens? Please feel free to shoot those over. So that should pretty much cover it for today. And again, hopefully that was all relatively clear. Sorry about the slide snafu in the beginning. And that should basically cover it. Thank you all very much.